Hello everyone. Today we learn about neutrophil and its function. Neutrophils are most abundant white blood cell among all in circulation. They are the first line of defense in innate immunity. Their main function is capturing and destroying invading microorganisms through different mechanisms like phagocytosis and intracellular digestion, release of granules, and third is formation of NET, neutrophil extracellular traps. Neutrophil life cycle begins in the bone marrow with granulopoiesis. Granulocyte differentiation is regulated by the myeloid transcription factors. Mature neutrophil contains granules and secretory vesicles containing specific proteins. These granules are formed at the different stages of maturation. Like primary granules, also called azurophilic granules, they are found at the myeloblast and promyelocyte stage. Secondary granule, also called the specific granule, they are found at myelocyte and metamyelocyte stages. Tertiary granules, also called gelatinase, they are found at band stage. Secretory vesicles are the only detected in the mature neutrophil, and granules contain antimicrobial enzymes like elastase, myeloperoxidase, cathelicidins, defensins, matrix metalloproteinases, etc. The neutrophils are continuously produced by the bone marrow. They are entering into the circulation and migrating to the site of the need and eliminated by the macrophages. This cycle completes within a day, and this is a continuous cycle. Mature neutrophils migrate through sinusoidal endothelium. And endothelial homeostasis is regulated by the vascular endothelial cadherin. The production of mediators like macrophage inflammatory protein 2, MIP2, GCSF, CXC, motive chemokine 1, leukotriene B4, complement 5A, these all stimulate the release of mature neutrophils. Neutrophil circulating in the blood is called the circulating pool of the neutrophil. And other pools are bone marrow pool, when neutrophil receives the anti-apoptotic signals in the bone marrow. Other pool is marginated pool. They are loosely adhered to the endothelium of the venous blood vessels. And the last is neutrophil reservoir in the different organ like liver, spleen and lung. In infection or inflammation, neutrophil migrate quickly from peripheral blood into the site of infection or the inflammation. This process is called extravasation. Inflammatory reactions are generated and influenced by the inflammatory mediators like chemokine, peptide, eicosanoids, etc. These mediators are released by sensitive leukocytes like macrophages, dendritic cells, mast cells, by endothelial cells, by epithelial cells, by fibroblasts, and by activated neutrophils. Chemokines are the group of the chemotactic cytokines, and they are divided into the four groups: CXC, CC, C and CX3C according to the arrangement of the 2 and terminal cysteine residues, where X stand for amino acid. CXC, these chemokines target mainly neutrophils and lymphocytes. CC chemokines target macrophages, eosinophils, basophils, dendritic cells. Certain surface structures found on the pathogens, also called the pathogen-associated molecular patterns, PAMs, in case of infection, and endogenously released surface structures after the cell damage, also called the damage-associated molecular patterns, DAMPs, in case of cell death. They sense them through the receptors like pattern recognition receptors, PRR. This in turn releases the pro-inflammatory mediators like interleukin 1b, interleukin 6, TNF alpha. This triggers the accumulation of leukocyte at the site of infection, inflammation or injury. This also produces acute phase reactants and regulate the vascular endothelial permeability. Recruitment of neutrophils at the site of infection or inflammation needs the adhesion and transmigration through the vascular walls. In all tissue, migration occurs in the post-capillary venules, except in the lung, where this process occurs in the capillaries. This is the graphical representation of the rolling, adhesion, arrest and crowding of neutrophil at the site of infection or inflammation. The first step is activation and upregulation of adhesion molecule in the post-capillary venular endothelium at the site of inflammatory tissue. Important adhesion molecules are P-selectin and E-selectins. P-selectins are stored in the endothelial cells and the platelet, and after activation, P-selectin reaches to the apical cell membrane of endothelium. E-selectin is de novo synthesized at the endothelial surface. These both bind to adhesive ligand present on the neutrophil. Usually cellular components like neutrophils and other white blood cells located in the center of the small blood vessels while blood is flowing and where the flow velocity is maximum. In inflammatory foci, blood vessels are dilated due to the effect of inflammatory mediators. This results in the decrease in the flow velocity. This enables neutrophils to bind endothelial surface. However, due to the velocity forces of the blood, cells cannot remain anchored at the surface of endothelium. The cell is rolling on the endothelial surface by constant making and breaking contact with endothelial surface and the cell. The next step is G protein couple receptor on this rolling granulocyte bind to the attractants secreted on the apical membrane of the endothelium. This leads to the conformational change in the granulocyte surface protein termed as beta-2 integrins. The important beta-2 integrins here are lymphocyte function associated antigen 1, LFA1 and macrophage antigen 1, MAC1. They both interact with the intercellular adhesion molecules ICAM1 and ICAM2 on the endothelial surface. Due to the conformational change, the granulocyte now firmly bind to the ICAMs, causing the end of the rolling mechanism, and the granulocyte is adhered to the endothelium, and this is called the arrest. After this arrest, granulocyte crawl to the endothelial passageways. This crawling is due to the firm binding of MAC1 to ICAM1. This leads to the migration of granulocyte perpendicular to the endothelium and they reaches to the site of need. These recruitment mechanisms are not always present in the all organ. Example is in the liver, the rolling mechanism is absent and neutrophil adhere directly to the liver sinusoidal endothelium by interaction of CD44 on the granulocyte and hyaluronan on the endothelium. After the completion of transendothelial migration, neutrophil migrate through the interstitium along with the chemokine gradient. Granulocyte use the amoeboid mode of the locomotion to reach the site. This process is characterized by the actin-myosin contraction within the cell and the activation of beta-1 integrins. Neutrophil can detect the extracellular chemotactic concentration gradient and move towards the higher concentration. This whole process is called the chemotaxis and it has three different processes: gradient detection, polarization and cell mobility. Chemoattractants can bind to the proteoglycans of the extracellular matrix and thus they form the concentration gradient. The neutrophil have the receptors for the chemotactic molecules like complement factor 5A, leukotriene B4, CXCL8, an exogenous molecule released by the bacteria. These receptors are linked to the G protein receptor signaling pathway, which leads to the conformational change and cell undergo the polarization. These form the front end and the rear end of the cell, and neutrophil integrins are activated and directs the cell to the higher concentration gradient. Constant presence of neutrophil in the tissue leads to the tissue damage and the chronic inflammation. Neutrophil must be cleared away from the injury site after the role is done. The neutrophil clearance is not solely dependent on the macrophage removal mechanisms. In some studies, it is seen that depletion of monocyte or macrophages did not compromise the neutrophil clearance. And also, few studies have noted that the neutrophils are able to undergo bidirectional movement, and it can go from the extracellular matrix into the blood vessels, and this process termed as a reverse migration. Now we discuss the functions of neutrophil at the site of action. These are phagocytosis, oxygen dependent mechanism, oxygen independent mechanism, and NETs, that are neutrophil extracellular traps. The first function is phagocytosis. It is the first line of immune defense is the form of receptor mediated endocytosis known as phagocytosis. The foreign particle and the altered cells both are eliminated by the phagocytosis. The neutrophil phagocytosis is a rapid process and can be completed within few seconds. The foreign cells like bacteria are labeled by the antibodies, and the complement systems are known as opsonization. So binding of the phagocytic receptor to the correspondent ligand induce the clustering of receptor on the neutrophils, and following the signal cascade that result in the actin dependent formation of phagocytic cup. The elongation of the pseudopod occur around the ligand, and internalization of the target occurs in the form of the vacuole. It is also called the phagosome. Finally, particle is degraded and microorganism is eliminated. Other mechanisms beside the phagocytosis are oxygen dependent and oxygen independent killing. In oxygen dependent mechanism, NADPH dependent production of superoxide ions occurs, and they leads to the formation of reactive oxygen species like hydrogen peroxide, hydroxyl radicals, and hypochlorite acid. This acid leads to destruction of bacteria. Second is oxygen independent mechanism. It is a degranulation. The degranulation of the visible granules of the neutrophil is important mechanism, and these granules are subdivided into the three classes according to the content. Azurophilic granules, they contain antimicrobial substance, lytic enzyme, peptides, defensins, and myeloperoxidase. The specific granules contain phagocytic receptors, NADPH oxidase complex. A patient who lacks the specific granules, they are susceptible to the repeated skin and respiratory infection and defective chemotaxis and the adhesion. The tertiary granules, they contain the receptors and the enzyme to degrade the extracellular matrix that facilitate the extravasation and the migration of neutrophil at the site of inflammation. The third mechanism is formation of neutrophil extracellular traps, NETs. At the end of the completion of the lytic process, the nucleus of neutrophil is released as a net-like DNA structure into the extracellular space, and this is called the NETs. They contain the histones and cationic peptides. After release of NETs, they can surround, immobilize, and kill the microorganism including bacteria and fungi. Neutrophil functional response depend on the transcriptional activation and the by change in the expression of the surface molecules, and this leads to the occurrence of the different phenotypes of the neutrophil. Thus they form the various neutrophil subset, also called the subpopulation of neutrophils. Half-life of neutrophil is around 6 to 12 hours, They remain in the circulation for few hours before they leave into the tissue. During this time, they are changing their phenotypes. Changing in the phenotype occurring when the neutrophil leaves the bone marrow until they leave the circulation, also called the age neutrophil, in the absence of inflammation, is called the aging. These aged neutrophils are acting as an activated neutrophil similar to the neutrophil at the site of inflammation. Neutrophil in the different tissue perform specialized function depending on the organ. Examples are in the lung, the large number of neutrophils accumulate and adhere to the vascular lumen and the interstitial space. In spleen, many neutrophils found in the marginal zone producing cytokines and that simulate the antibody production by marginal B lymphocyte. In lymph nodes, they interact with the T lymphocyte and mediate the T cell activation. Some subtype of neutrophils are capable of stimulating angiogenesis in the non-vascularized tissue under the hypoxic condition. These all are the physiological subpopulation of neutrophil. Now we learn about the subpopulation in the disease. Neutrophil subpopulation induced by the infection of bacteria. The microbial products derived from the microbiota like endotoxin can influence the neutrophil aging and development of the B cell helper neutrophil in the spleen. Subpopulation in inflammation. We have already discussed the first line immune defense by the neutrophil at the site of infection or inflammation. Now here we are discussing about the obesity where the inflammatory state is seen and where neutrophil increase rapidly and metabolic imbalance is generated. The activated neutrophil first release the elastase enzyme from the eosinophilic granules and this enzyme destroy the insulin receptor substrate 1 in adipocytes and in hepatocytes and this leads to the insulin resistance and the lipogenesis. So the neutrophil has a important role in the metabolic syndrome and diabetes. Subpopulation and the NET formation. The NET formation is seen in the several processes like atherosclerosis, venous thrombosis, inflammatory conditions like gout and pancreatitis. Now we discuss about the neutrophil subpopulation in the cancer. They play the important and contradictory roles in the cancer development. In the patient with the advanced cancer, the counts of the neutrophil in the blood are increased mostly due to the secretion of cytokines by the tumor and this generates the more granulocytosis. The presence of the increased neutrophil count in the peripheral blood is associated with the poor outcome in several types of the cancer. The presence of neutrophil in the tumor itself is an indicator of the poor outcome. So for the prognosis of the cancer, NLR, neutrophil lymphocyte ratio is important. However, the high NLR does not associated with the poor outcome in the all type of cancers. Example is in the gastric cancer. High NLR is associated with a good prognosis. Myeloid derived suppressor cells MDSC. Increase in the immature myeloid cells are seen in some type of cancers along with the increase in the neutrophil. These immature cells are at the various stages of maturation and differentiation and they accumulate in the spleen and they present the immunosuppressive phenotypes. So they are named as a myeloid derived suppressor cells MDSC. Another subpopulation is low density neutrophils. When neutrophils are purified by the fecal density gradient, they appear at the bottom of the tube at the high density fraction. But a subpopulation is identified at the low density fraction and this low density population increases with the tumor growth and the progression. They are also seen in the conditions like SLE, rheumatoid arthritis, rheumatic fever, sepsis, psoriasis, HIV infection, asthma, malaria, ANCA associated vasculitis, and also in the natural pregnancy. During pregnancy, the maternal-fetal tolerance is important, and for that, the downregulation of the T cell function is must. The arginase enzyme depletes the L-arginine, which is required for the T cell function. The source of this increased arginase activity is a low density neutrophils. Tumor associated neutrophils. The subpopulation of the neutrophil during the tumor progression is related to the infiltration of neutrophil into the tumor. The infiltrated neutrophils have two types of subpopulation called N1 and N2. Tumor associated neutrophil can show the anti-tumor also called the N1 phenotype or pro-tumor also called the N2 phenotype depending on the tumor microenvironment. They are described only in the murine models of the cancer and the nature is largely not known in the human cancer. So this population need to be studied in the depth in the future. Immunogenic cell death. The immune system detects the altered cell molecules in the damaged cell through the danger signals or the alarmins. These are referred as a damage associated molecular patterns, also called the DAMPs. These stimulate the neutrophil to utilize the cytotoxicity against these damaged tumor cells, and this is called the immunogenic cell death. It is now known that the neutrophil functions are more than just a phagocytosis. They are transcriptionally active complex cell. They produce cytokines, modulate the activities of the neighboring cells and contributes to the resolution of inflammation, and they actively functioning in the several diseases like cancer. There are currently no appropriate molecular markers available to identify these all different type of the subpopulation. And these all subpopulation are the circumstantial evidences and experimental proofs are needed. So all this population need to be studied in the depth in future. These are the references from which I have taken the content and few photographs. This is about the neutrophil and its function in the immune system. Thank you for watching.